Thanks for joining us for another edition of Biotech and Life Sciences CEOs as we focus on leaders in Atlanta in the life sciences community. Today, I am truly honored to have as our guest Dr. Dennis Liotta. Dr. Liotta is a world-renowned medicinal chemist, an innovator in drug discovery, particularly in the forgotten diseases which afflict the world's poor. The Samuel Candler Dobbs Professor at Emory University and an important philanthropist. Dr. Liotta holds over 100 patents in the US, with, I understand, an additional 20 on the way, including in Entriva, which is a component in many of the combination therapies for HIV and PrEP, as well as Epivir, the first FDA-approved drug to treat hepatitis B. Both of those drugs are on the WHO list of essential medicines. In addition to his creativity as a research scientist, Dr. Liotta has co-founded more than 10 biotech companies, among them Pharmasset, which developed Sovaldi, the blockbuster curative hepatitis C drug, and that was acquired by Gilead Sciences in 2012. Dennis earned his BS in chemistry from Queens College in 1970 and his PhD from City University of New York in 1974. He did his postdoc at Ohio State and has authored more than 300 peer-reviewed publications. He's been honored as the Thomas Jefferson Award winner in 2011, Emory University's highest service award, as well as the 2022 Perkins Medal winner, generally regarded as the highest award for achievements in applied chemistry. Welcome and thank you for taking the time to join us today, Dennis.
Thank you, Tim.
So how is it, Dennis, that you chose academia rather than going to work at a major pharma early in your career? And then what caused you to turn the page later in your career to commercial development and entrepreneurship?
So to the first question, it started when I was very young. As a matter of fact, about 9 years old. I had an older brother, 12 years older than me, and he was starting graduate school in chemistry. And and then he later went on to have an academic career. And my brother Charlie is quite a remarkable person. He could take the most complex concept and explain it in a way that makes you at least feel like you understand what's going on. And but I was always in awe of that. And I said, I want to be like him. I want to be able to do that. And what better venue for that than an academic institution where every year you get a new crop of students who come in and who challenge you, and a place where you can select whatever research you want to do. You can get invited to conferences and travel. It's a wonderful environment, and they even pay me, not as much as I would like, but they pay me too. So this is it's an opportunity. It's like a blank canvas that you can fill in any way you want. So I love academia. I love the students, and I love the opportunity to be creative.
Okay. And then how did you turn the page to
Yes.
entrepreneurship? Why?
So it's very interesting. Also, my life is it's a bit of an anomaly going back a long ways, and I always tell people, um, don't don't use me as a model for how you're going to go forward, because my path was always very circuitous. In my particular case, for the first 12 or so years of my academic career, I was doing what we call organic methodology. So we were looking for new ways um, to do important chemical transformations. What defines importance? Whatever I thought was important, right? But usually in organic chemistry, if there's a way of forming a new way of forming a carbon-carbon bond, that is potentially of some importance. In the mid 1980s, I started uh, consulting at some pharmaceutical companies, and that was my first exposure to medicinal chemistry, and I liked it. I hadn't had a biology course since 10th grade in high school. I
Wow.
never really took much biology in college. I was a chemistry geek, and so so I didn't have this very robust background in biology. But by 1988, I felt that the AIDS crisis had gotten out of hand, and most pharmaceutical companies were shying away from the disease. I think there were a couple of factors involved, and I think the social stigma part of it was real, but the more important part of it that you can understand, Tim, was the market size. It was uh, by pharmaceutical standards, it was a small market size, about $250 million a year. And so they said, well, the market's too small. And I had a chance to talk to several pharmaceutical executives, and I asked them, why do you think the market's small? And before they could answer, I said, perhaps it's because people keep dying. If we could find drugs that could keep them alive, then they'd have to take those drugs every day of their lives. And we could turn it from a small market to an enormous market. And they said, yeah, well, we'll see if we can do that. But so I said to myself, look, I don't think these guys are putting in the appropriate effort to really make this happen. And I couldn't do any worse than they could do. And so I said, you know, I'm a pretty good organic chemist. Let me see if we can figure out some good ways of making some of these compounds, and then see what happens. So, so that's again a kind of crazy way of getting into it, but I just felt personally compelled. Some of the most creative people I knew were dying, and somebody had to do something.
So it was personal for you,
Absolutely. Absolutely.
and maybe that's why it worked.
Maybe, there was some serendipity involved. Um, I I was in the right place at the right time, but Pasteur is famous for saying chance favors the prepared mind. So
The,
some combination of things.
the prepared mind. But and so as you began that process, did you think at that time that there was something? What made you think that you really were the one that could make an important contribution? How did you begin? And
Well, it's very interesting. So I knew very little virology. As I said, I was a pretty good chemist. So I knew I had to find someone to partner with that knew about virology and something about drug development. And so I was introduced by a former student to my colleague Ray Shinazi, and he was part of a virology operation.
Okay.
And our personalities were about as different as one could find, but he was the person, he was going to be the best chance I had of making a difference. And so I said to myself, well, we've got to find a way to make this relationship work. And we did. And we became good friends. And as I said, his style is different than mine, but somehow we got it to work.
Okay. And then, so these drugs are taken in combination. And so I'm interested in how that sort of, I guess, collaboration, not you and Dr. Shinazi, but you with the makers of other drugs in the combination. How did that come about and grow? Was it the NIH or what happened?
Yeah, so I was fortunate to get some good support from the NIH that allowed my lab, which was very tiny, to begin to grow. Um, by the way, I had an interesting little sidelight. One of my senior colleagues in the department, I told him, I think I'm going to get into HIV drug discovery. And he said to me, I always thought you were pretty smart, but hearing this, I think you're either stupid or crazy. He said, when you worked in organic methodology, he said you were competing against other academic labs. He said, now you're going to compete against giant pharmaceutical companies that have hundreds of talented scientists and infinitely more resources than your lab could ever hope to get. They're going to bury you. And, you know, his advice was legit. I had to admit, it's one thing to do something noble, but noble could also be stupid. And so it turned out, again, the serendipity portion of it. Raymond went to a conference in Montreal, an international AIDS conference, and he there was a poster by a Canadian company on a compound that showed some good potency, and they said it wasn't very toxic. And that was really important because all the other drug candidates that we had at the time were very toxic. And there were big problems with that. So a long story about how we got into this series, but I resisted because I said, look, they've obviously patented this stuff, and so what are we going to do that's going to be novel that we could anyhow? We uh, we started to look at some methods for making it, and we found a a fabulous way of making these compounds, just infinitely better than the chemistry that was out there. And I said, well, if nothing else, we can assist uh, in the manufacturing of this thing, which was silly. I showed my naivete. Anyhow, this enabled us to make a lot of novel compounds that others could not do with the existing chemistry. And that's really how, for example, Emtriva came about, because we made this compound and we tested it, and it had a beautiful profile. It was really spectacular. And the more we tested it, the better it looked. And compared to other drugs that were out there at the time, this looked like it was the one. And Epivir, which you also mentioned, was a close second. And so we thought, wow, these are great. And so that's how we started working in there. And it was because we came up, I had a really talented postdoc, and he he got this chemistry to work beautifully. And usually when we do chemical reactions, we get our product, but we have to spend a lot of time purifying it from byproducts. And the first reaction in this series that we did, he came and showed me this NMR spectrum, which is one of the tools we typically use to identify the structure, but also get an idea of how pure it was. And it was a beautiful spectrum, nice and clean. And I said, wow, that's great. I said, how did you purify it? He said, I didn't. This is the way it came out of the reaction. So I said, okay, we've got something here. And we jumped on it, and we were able to make a lot of compounds quickly. And then we had some other hurdles. I don't know how technical you'd want me to be, but maybe I could give you a very brief idea.
Yeah, please.
So when we first made these compounds, the chemical term that applies is they were racemic. They were so equal amounts of two non-superimposable mirror images. Now that sounds very intimidating, but maybe I can use a couple of props to show you what I mean. And I always carry these props with me. So my hands. So these are two mirror images of each other, but they're not superimposable on each other, right?
Right.
And if you take the analogy a little bit further, if you have a nice snug fitting glove, um, the left hand will only fit into that left glove comfortably, and it'll be really struggle to get it into the right-handed and vice versa, right? So this is the same as what happens in vivo. One of these mirror image compounds will bind much better to the biological target than the other. So we had to figure out how to separate these two. And we decided to take a clue from nature. Nature uses enzymes to differentiate between these two mirror image compounds. And we found an enzyme that would do a chemical reaction 1,000 times faster with one than the other. And that, again, without getting too much in the weeds, that enabled us to make and separate and get these compounds cleanly, because that's what the FDA wants. They don't want you, one of the other isomers might be considered a 50% impurity, and FDA certainly doesn't like that. So we were able to innovate all this chemistry and get these compounds in ways that nobody else at the time could. Others subsequently found ways of doing it, but we were the first, and that's how come we were able to have robust patents on these subjects.
Wow. When that happened, Dennis, when the postdoc came to you and said, look at this,
Mhm.
how'd you feel?
I was stunned, but elated. And elated is almost too uh it my feelings were just I couldn't believe that this was we were able we had a hypothesis of why this would work, but most of the hypotheses that scientists make turn out to be wrong.
Right.
Because and so, but this was, it wasn't like it was pretty good. This was just outstanding, right? And so I I said, oh, thank God, but we got something here, and I don't know where it's going to go, but I'm going to try and make something.
Well, I always think that success follows the person that takes the endeavor personally.
Mhm.
And
Yes.
that's characteristic of what happened with you. So you're still, I think, working in the lab on HIV. You're trying to abate the effects of the nephrotoxicity that can come from long-term use.
Yeah, there are some of the agents, and it occurred to me, you part of your question that you asked, I didn't answer, and that was, how did I get to all the other companies who
Right.
had different drugs, and how did we find a way to come together? And the amazing thing is, then and probably now, since we have to do things in combination, no one knows a priori which combination will work.
Okay.
Now we have some better tools that can give us some strong indications, but in the 1990s, we had no idea. You had to make them first and 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 test them. And and so partnerships developed around the resistance profiles of the compound. So once you got up and you presented your data at a conference, you published your work, then people started looking for synergies. So they would seek you out, or you would find the right kinds of partners who could do that. Everyone wanted to develop their own combination therapies without partnering with another entity, but many times the most expeditious path forward in the short run was to partner with somebody else. So those things kind of sort themselves out. But this issue of resistance, I don't know if your audience is going to care, but so Emtriva, which you mentioned, is still first line therapy for HIV. It's in so many different combinations, and yet I told you what a fabulous drug it is, but it had one flaw, almost fatal flaw, and that is that it if you give it as a monotherapy without anything else there, it develops resistance very quickly. What does resistance mean? In this particular case, it would take 500 or 1,000 times more of the compound to address this single resistant mutant. And as you can imagine, that is not viable in drug therapy. You can't suddenly give um, 10 times the dose to overcome that. So that's where the synergies came in, and AZT was the first drug approved for HIV. And because it was the first, it had to be a monotherapy. But it was a quite a dirty drug, a lot of side effects.
Mm.
And like every other HIV drug, over time people developed resistance, and then the drug was no longer effective. Um, and I don't know if people care about this concept of resistance, but it's not very hard to understand. Um, HIV, one in every 10,000 replications, we see an error or a mutation, and viruses don't have any mechanism for for correcting these errors. So whenever an error happens, it's just there, and So 1 in 10,000, there's an error, and there are 1 trillion 10 to the ninth replications per day. All right, so you could imagine that even in a day, we get a whole series of mutations. Now, some of them are just die out, they're not very viable. The ones that are viable will respond to drugs differently. So when they respond well to the drug, then those are suppressed. We don't see them. But there's always some outliers that don't, uh, the drug just doesn't fit. It's like that glove again, where now we've changed the glove shape a little bit and it doesn't fit in there as well. Those just grow up, and then after a while, uh, so so that's what happened with Amtriva. Just one mutant caused this problem, and it turned out colleagues both here at Emory and at what was then Burroughs Wellcome, now part of GSK, um, found that the combination of AZT and the one of these drugs, emtricitabine or epivir, um, they were AZT was 50 times more sensitive to this one mutant than um, than it was to what we call the wild type virus, the most evolutionarily favored um, strain of the virus. So that meant we could give AZT in combination with Emtriva or Epivir, but we only had to use 1/50th of the concentration. So all those nasty side effects from AZT were just much less because we're giving it at a much lower dose. And then these other drugs that we found, we could give them, they can control everything else. So it was the first actually rationally designed combination therapy for HIV, or maybe for anything. I'll put a question mark on the anything, but in HIV, our drugs were, this was the first rational combination therapy, and that changed the way we thought about treating uh, just viral disease in general. That is, if we understand the resistance profile and we get drugs that are complementary to each other, we can put them together and they can suppress replication and across the spectrum of mutants. But I don't know if that's just too uh, too geeky to
No,
talk
not at
about.
all. In fact, that I wanted to ask you, I saw something in, I think it was in Stat maybe recently,
Uh,
um, about what they called the PrEP trial
mhm,
in Africa, which had been stopped because it was determined that there wouldn't be much chance of success. But I know that Moderna and the NIH and the International AIDS Vaccine Initiative are working on some sort of germline targeting approach for a vaccine. Where the participants are given several shots, kind of hoping to nudge the immune system into creating the perfect antibodies.
mhm.
Do you think there will ever be an HIV vaccine? Can we get there?
So I pray there will, but I'm um, hesitant to say that it might. And let me also try and give you a very simple idea. So uh, when COVID happened, right, we um, companies jumped on it, and we had a vaccine in a year or so,
Right.
right? Remarkable. That they need to be applauded. And the one of our new Nobel laureates uh, you know, was responsible for that, and that's great. Those have a problem in HIV, and the problem is not that hard to understand. So vaccines work by creating antibodies that can sample something on the outside of a virus that it will recognize, and then it binds to it, and that marks it, that particular virus, for destruction by another agent, right? But these surface receptors are hyper variable. They're changing all the time. So when antibodies come along and they sample these, they'll or the B cell will come along and make a factory. But when the right cellular agent samples it, then it starts a process that says to the immune system, let's start manufacturing antibodies. But that process can take 4 or 5 days before the antibodies are present in sufficient quantities to be viable. In that time, those surface receptors on the virus have changed. So we call those things escape mutants, and escape mutants form more rapidly than the antibodies can be made. So you're chasing a speeding car down the road, and you're losing pretty much just because of the time it takes for the immune system to generate enough antibodies to do that. So that's my reason to think that uh, unless we have some new innovation that can stop that process early, we might have a problem. Now, these messenger RNA based vaccines have been remarkable, and it's possible they can find some innovative ways of doing that, but that is the fundamental problem why we've gone 40 years without developing an HIV vaccine.
So I'm not sure, aside from just the method of transmission, if there's any commonality between HIV and Hep C in terms of the virus itself. Is there some way that your work in HIV kind of informed your work in Sovaldi? Is there a connection?
Yeah, so yes, so there were similarities, and of course there were differences. So both HIV and hepatitis C, and even hepatitis B, are the method of transmission is exchange of body fluids. The body fluids in hepatitis C could come from sexual intercourse. They could come from blood transfusion. They could come from use of a dirty needle, so just intravenous transmission. There are a lot of ways, but fundamentally they all involve a transfer of body fluids. With Sovaldi, what we did was quite, I thought, interesting. Back in 1998, when we started Pharmacet, nobody had any ideas about a drug for hepatitis C. And part of that was there weren't any really good assays available. And it wasn't until 1989 that hepatitis C was actually identified. We knew it was there. Yeah. But for years, if you went to conferences, they called it non-A, non-B hepatitis. which which I think is very telling, right? Because it says, look, there's something there, and we know it's in the blood supply because people are getting this infection, but we don't know what it is. And then some very clever virologists identified it, and about 4 or 5 years ago, they three of them received the Nobel Prize. So we finally find out what it is, and we finally start to develop tests that can reveal when blood was contaminated. But up until then, there was a generation of people who acquired hepatitis C through no fault of their own. They just went into a hospital, had to get blood, and it was contaminated. Um, so once we knew of what hepatitis C was, it turned out that it was an RNA virus, a single-stranded RNA virus. And so we knew what kind of, in a general sense, what kind of nucleic acids would be the substrates for that. And just like we did in HIV, we spent some time in the couple of years developing some methods for making the right kinds of agents. And then people started to develop models where we could test our compounds, and they created an assay they call a replicon assay, which is essentially a virus that is engineered so that it can't replicate. So it was safe to use, but it had just some of the machinery missing, but otherwise it was a pretty good model. And once we had that, we could crank through compounds pretty quickly because we had all the experience of making these so-called nucleoside analogs from our prior work. And again, I'll go back to what my colleague said, right? He said, you're going to get buried. You're never going to be able to do this stuff. But we beat them to the punch. All our competitors, they got to it later. Now they had, again, a lot more people, but by 2003, we had essentially the nucleoside that is Sovaldi in hand, and it wasn't didn't have all the properties that we wanted, and that's where the Pharmaset guys um, they they we in order for it to be activated, it had to be phosphorylated, had to add three different phosphate groups on in order for it to be active. And that it turns out the cells didn't, that first phosphorylation step was very inefficient. So the guys at Pharmaset made what we call a prodrug, something that already has the phosphate in there, uh, but you couldn't just use the phosphate by itself because it's charged, it can't get into cells. So they put some groups around it that metabolized off intracellularly to give the right compound. And once that blockage was passed, then it went from an okay kind of compound to the blockbuster that changed the face of the world as far as hepatitis C goes. So we in kind of came up with we again, Raymond Chinazi and I and the people we worked with came up with the right nucleoside, and then the Pharmaset guys, um, they did that second very important step to get this to be a really outstanding compound. some very talented people at Pharmaset. Um, and that's when Gilead said, okay, this looks like it's good. And it was one of the few drugs that could be effective as a monotherapy. So the resistance didn't develop that fast. And liver cells are kind of special. Liver's about the only organ in the body that can regenerate itself.
Right.
And so, and Gilead bought it for $11.2 billion. And in 11 months, made that money back from sales.
Right. That's right. They did. So,
So,
Pharmaset was sold to Gilead, as you say, and kind of much of our audience are founders.
mhm.
So for their interest, can you tell us about kind of how the transaction came about? Were you actively involved in it? Um,
I
do you
was,
have any advice for a founder approaching that sort of transaction? Probably not 11 billion, but something that
yeah,
would be significant.
I was not involved because um, initially, of course, I started out on the scientific advisory board, um, and the company evolved, and they Raymond and the management of the company had some disagreements. If many of your people in the audience are founders, they won't be surprised that people can disagree about the way to go forward, and um, so he basically cut his ties with the company and said to me, um, because I was getting along fine with him, and he said, "Well, look, you can't you can't do both. You can't collaborate with me and collaborate with Pharmaset. You have to make a choice." So I had a long-standing relationship with Raymond, and I said, "Okay." So I wasn't after 2005. I wasn't really involved, and if I said I was involved in the transaction, then I'm sure the SEC would come knocking on my door and say, "Well, wait a minute now,
Okay. Okay.
did you ever hear of insider trading?" And
Right. Right.
so I want to be very clear to any folks in the SEC that I had no involvement whatsoever. But it was some of our colleagues at Pharmaset who are very good. One of our Atlanta colleagues uh, uh, he, was the VP for business development at Pharmaset, Dr. De La Rosa, and he's he was very good at pitching, and of course, he had a good product to sell, which helps.
Right.
So
It starts with a product. So one of the things I guess you're working on now is an antagonist for a protein receptor, which might have value in terms of the treatment of multiple cancers. Can you tell us something about that and how you got involved?
yeah, so we have actually several projects, but I think you're probably referring to the CXCR4.
Right.
There are almost 50 cancers that overexpress this on the surface, um, and these tend to be difficult cancers to treat. They don't, for example, respond well to many of these checkpoint inhibitors that have become
Everywhere.
very, you know, they've in the last decade, they've changed um, cancer therapy because they're immuno-oncology agents. They use the immune system. So Merck has Keytruda, um, uh, Bristol Myers Squibb has Opdivo, and there are a bunch of these that interrupt a certain kind of handshaking interaction, and they work well in certain types of tumors and not at all in others. And it appears, to put it on a simple level, that the tumors that it doesn't work well, we call cold tumors, and the ones that it works well in, we call hot tumors. So you can see how simplistic this might well be.
Perfect
And
for me.
but it's the cold tumors tend to be ones that are heavily infiltrated by immunosuppressive cells. So immune cells that that are used to keep the right balance in our immune system, prevent it from getting too revved up. So the immunosuppressive cells shut down an immune response, and for some reason that is not well understood. These cold tumors are heavily infiltrated with immunosuppressive cells, and the effector cells, the ones we'd like to get into the tumor, really can't um, uh, permeate in. Uh, so these CXCR4 antagonists um, have this unusual property that I'll spare you the complex details of, but when they bind to the surface receptors on the tumor cells, they cause these immunosuppressive cells to egress from the tumor, and that then makes it accessible for effector cells to permeate in and do their thing, kill the tumor. So this is a work in progress. The hypothesis has been validated, and we're trying to see if we can. So it's been validated in animal models, right? But we're trying to see if we can put something together that we can make the strong case that it will work in humans, because these tumors, there really aren't good treatments for any other way. So we're trying to basically create an adjuvant to the checkpoint inhibitor that, when used together, will heat up cold tumors.
Okay. Okay. One more thing you're working on, and I want to get to your work in Africa here as well, but something I'm personally interested in is that you're working on something called NMDAR receptors, which negate the decreased response over time of the analgesic effects of opioids. And therefore, those would cause the user to seek more and more to kind of get the same effect. Can you tell me about that? And aside from maybe the tragedy and the of the addiction and the overdoses that have permeated the society, was there a scientific driver for your involvement, or was it more like AIDS? You were looking for a solution to this massive problem.
Yeah, well, it's always a little of both. I just, as I guess, one very brief historical anecdote. I told you when I was getting ready to make this transition to medicinal chemistry, um, but hadn't flicked the switch yet. I went to a pharmaceutical company, and the standard thing one would do is you give a talk, and then you spend the rest of the day talking to the scientists there. And I met this one woman who was very, very accomplished medicinal chemist, and she said, "Nice talk." And I said, "Well, thank you." And she said, um, "It's very good work, but it's a problem in search of a solution." Oh, no, excuse me, I said it wrong. A
Solution is
solution in search of a problem. And I was stunned. She said, I don't mean that in any kind of insulting way. It's just not what motivates me. I want to solve problems in the health of the public, and I have to be focused. I don't have the luxury of just developing these solutions and looking around and hoping that someday someone will come along and find the problem that they can be applied to. And that really stuck with me, right? I said, yeah, do I want to look back 30 or 40 years and say all I did was create these interesting little things that in essence were esoteric, that maybe a dozen people in the world would say, well, that was really good work. That was so so we I this idea of being impactful was always a driver. And we use any source of information we can. I have a great collaborator in our pharmacology department, Steve Trinellis, who's an expert in the NMDA receptor. And it's a post-synaptic receptor with a lot of different subtypes and all sorts of interesting combinations. Interesting from an intellectual perspective, maddening from someone who's trying to develop drugs, because you never quite know exactly what you're dealing with. But having Steve as a collaborator has been great, and there are various things you can do to either inhibit, antagonize the process, or to potentiate, to amplify, depending on whether the function you're looking at is either something that's less than you would want it to be, or it's too much and you want to turn it down. So we develop both antagonists and potentiators to be able to address different kinds of situations. And then we also will take whatever else comes along. So there, for example, there are a lot of natural substances that we call neurosteroids. They're steroids that are operative in the brain and will have functions. And um, like many steroids, these things are kind of they don't have drug-like properties because they weren't evolved by nature to be a drug. They were there to cause some specific neurological function. And so we figured out ways of modifying them to maintain the activity, but make them better. And we use whatever we can get. So you may have heard of magic mushrooms, and the active ingredient in magic mushrooms is a compound called psilocybin. And what that does is it can remodel neural networks. They call it neuroplasticity. And and I'm just fascinated by this, instead of having someone take a drug every day, if we can remodel the neural networks, they may be able to take a few doses, and then the situation can resolve itself. Obviously, there's a long way between that hope and actually getting something, but it's in major depressive disorder. It's showing very promising results. So
Really?
so we do all of that stuff, and we're agnostic as to exactly which opportunity looks the best. We just kind of jump on the ones, where it comes from. We just jump on the one that we think is going to be the best. So.
Okay. Dennis, as a non-scientist,
Mhm,
the thing that fascinates me the most about life sciences is the people,
mhm.
dedication, the just plain off the chart brilliance, and especially the generosity. And you started a concept called drive,
Mhm.
which again is about the neglected diseases of the world's poor. And you're continuing your work in Africa. Can you tell us more about that work in Africa, where it's going, and what your hopes are for it?
Sure. Um, so I went to my first visit to South Africa was in the fall of 1999. And I went there with colleagues from Georgia Tech, Imperial College in London, and the University of Cambridge in the UK. And we had a big uh, contingent of people that went to South Africa to explore um, potential research and teaching collaborations. And in that trip, um, I fell in love with that country, I thought. And it's got plenty of problems, and God knows,
We do
uh,
too, you know.
yeah, but I think there corruption is probably still the worst problem, but it also had uh, the best medical infrastructure on the continent. The first um, heart transplant was done in Cape Town.
That's right. That's right.
Christian Bernard, right?
That's right. I
And
remember that. I was a kid.
so they had a lot of really good people, but in the area that I work in, medicinal chemistry, um, people that did that were mobile, right? They could go to Australia, they could go to the UK, they could go to the US, and they did because they didn't want to live in under the apartheid regime. So an entire generation of scientists who would have normally generated that second generation of people who could address these problems went away. And so I said, these are smart people. This is a great country. And there's got to be something that we can do to revive this opportunity because the possibilities are endless. And people talk about Africa in the next 50 years about what it might evolve to. Um, and yet it has enormous problems. And so I just got um, sucked into this idea that maybe we can help find some solutions. So HIV, we're doing what we can, and and I think our work and the work of our colleagues around the world have changed the face of HIV infection in South Africa. Uh, but in the process, non-communicable diseases were neglected, and one of the biggest is cancer. And a lot of people in Africa and in South Africa die from cancers. And if you think about the problem, you have these urban areas with pretty sophisticated healthcare facilities. But in the rural areas, there's really very little. There are a couple of local clinics, but they're not terribly sophisticated. So people in rural areas, if they need to get a 10-week infusion of a cancer drug, it's not accessible to them. They can't afford to go to the urban area. They can't afford to live in that urban area during that time period. And so there's very little that they can do. So we had this idea of possibly um, developing some um, orally active combination therapies. Um, and we thought we could use telemedicine techniques to monitor them perhaps through some blood tests, which could be give us an indication of whether our drug combinations were slowing the progression of the tumors. And so I just got back from South Africa last week, and I met with amongst others, the head of their regulatory agency. It's called SAFRA, and it's sort of their equivalent of the FDA, and to tell them this idea about this combination therapy that we think will be very safe, but will also combine a chemotherapeutic effect with an immunological effect, and that we can control the progression of cancers there, and finally have something that could work for these rural populations around the world.
Wow.
And I'll tell you one other funny story. So I was talking to a colleague in our Winship Cancer Institute who's an expert on healthcare disparities, and she's been terrific in terms of helping us understand the problem. And she said, I said, this is something that I think might really help South Africa. She said, well, if it works, it might help South Georgia too.
Yeah.
Because hospitals are closing and the situations aren't that radically different. The idea of either traveling every week back and forth to Atlanta when you're 2 hours away, or staying in Atlanta to get these treatments, it's not as extreme as it is in South Africa, but it's real. And so if we could find some new oral combination therapies that could be more effective, then we don't necessarily have to go 9,000 miles away to help people. We might be able to help people here too. That's a dream, but I'm a dreamer.
You are. Another of your philanthropic causes is encouraging the development of the life sciences here in Atlanta, creating an environment that would encourage young scientists to stay here in Atlanta as opposed to going off to Boston or San Francisco. Much like what you did with Drive in Africa. Can you share with us something about your vision for that and what you think might be the impediments, and how you think we can make it work?
Well, Tim, you know a lot about these kinds of activities. But let's talk about what we have and what we don't have. What are the necessary ingredients? Why did Cambridge, Massachusetts work? Why did South San Francisco work? Why did La Jolla in Southern California work? And they had the right combinations. So first, Atlanta definitely has the intellectual firepower between Emory and Georgia Tech and our neighbors not very far away, like the University of Georgia, the Mercer School of Medicine. And we've got the firepower. You know, there's no question about it. What we don't have here are a large number of funders, venture people, who, if you happen to be in Cambridge, their venture capitalists might be your neighbor down the street. And there also are a lot of scientists who are experienced. There are good laboratory facilities that are there all the time. Here in Atlanta, most of the real estate people don't want to build a lab because they don't know that it's going to be a sustainable investment. In fact, I've talked to people and they said, well, if you put in lab benches and fume hoods and everything, part of the contract will have to be, if you move out, you have to take those things out. and so instead of building infrastructure, people were reluctant. Now we have groups. I know you're familiar with the Portal group, and they're actually building facilities and they're providing the funding and the expertise to take this still nascent ecosystem and put those extra necessary ingredients in place so that we can finally um, realize the potential, the enormous potential that exists here. And you've heard me say this, but I like to use a one word descriptors sometimes because I find them to be impactful. So when you think of New York, you probably think of money. And when you think of Washington, D.C., you think about power. And if you think about Los Angeles, you'd say maybe glitz. And what is Atlanta? Atlanta is opportunity. We have enormous opportunities. We've got an amazing healthcare system here. We have um, uh, brilliant people, many of whom are quite entrepreneurial and are risk takers. And in a field like pharmaceuticals, where it might take 6 to 15 years to actually develop something and cost several $1 billion, you better be a risk taker. Otherwise, you'll get scared off very quickly. We have outstanding uh, intellectual property attorneys that can build the robust patents that we need that will give us uh, the wherewithal to span those times and still have enough time to not only make back the investment we put in, but will can finally generate those returns on investment. We've got a lot of super enthusiastic people like yourself, people who see the potential of Atlanta and want to try to um, actualize that. You know, in physics and chemistry, we talk about potential energy and kinetic energy, and that's what we want to take all that potential energy that's stored up here and turn it into something kinetic, something where we have viable um, infrastructure and expertise to take an idea. This is translational research from the bench top and translate it to the bedside, so that we can benefit the health of the public. And that's my mission. And my wife asked me when I was going to retire, and I ignored it for a long time. And then I told her, all right, I will retire at age 100. And after that, we can do whatever we want at that point. But until then, I'm gonna keep going forward.
That's great. Dennis, you're a man of the future. You're thinking about the the next drug you can do. But if I could just ask you for 1 minute to look back on your past with all the myriad successes that you've had, what makes you the most proud? What gives you the warmest feeling inside?
That's a very hard question because a lot of things give me warm feelings. Um, I could I could point to the HIV therapies which have proven so impactful. I could point to the success derived from Pharmasset. Um, but maybe something different might be all the incredible people who have come through my lab and who are now um, exhibiting their own creativity and finding new solutions. And I feel like we've created a large group of people who have been quite successful in the various endeavors, not necessarily in academics, but just in the whole arena. And I, I think maybe I'm most proud of them.
It's a way to sort of multiply all of those successes.
And just like viruses propagate, maybe creative scientists can propagate too. And I've tried and I think succeeded in instilling the passion in them, and this desire to make a difference. And I think they're going to do great things.
Dennis, you're there in your office or wherever you are today, and I'm here in mine. And as we conclude, I just have to say, I want to reiterate the same thing that I said to you when we met at the Emory Innovation. I just want to hug you, love you, and people like you because you're positive and you're making a change in the world. And it's just the greatest thing. So thank you for that. And thank you for spending some time with us today to explain a wonderful career. Thank you so much.
Thank you. And without folks like you who are trying to stir the pot and make this ecosystem work, it wouldn't happen. So I want to acknowledge the excellent work that you and some of your colleagues are doing.
Well, thank you. Thank you. Thanks for tuning in to the Life Sciences and Biotech podcast. We'll see you in the next episode. The information contained in this website and podcast are purely informational and not considered investment recommendations. Tim Doherty's participation in Biotech Insights is separate and apart from his role as an investment advisor representative. Nothing contained herein can be construed as a recommendation or endorsement of any of the companies discussed. Tim Doherty also has no financial affiliation with any of the companies mentioned in this communication. Tim Doherty makes no representation that the information contained in this material is accurate and is under no obligation to update this information as changes occur.